what I'm going to do today is give you a practical application of a graph analysis on a real psychological problem, specifically Autism Spectrum Disorder, or ASD. And the application I'm going to be using is IMaGES. Now, the NIH has estimated that at least 1 in 88 individuals has been diagnosed with autism. It's a very prevalent problem right now in the US, and it's something that seems to be growing. Actually, a few years ago, it was 1 in every 200. So whether that's a matter of being more sensitive to this kind of diagnosis or whatever, there does seem to be a problem, and that problem has brought a lot of research effort in trying to understand what's going on underneath this disorder. And the disorder is characterized by 3 main components. One of those is communication, and that's both linguistic and gestural. People with ASD have difficulties communicating. They have difficulty with emotional response and emotional regulation. And a third area that they seem to have problems with is behavioral responses. What are the appropriate behavioral responses for a given context? Now, the explanation for this is that there must be some sort of problem with the social cognition. Most individuals with ASD, and there's a wide range, by the way, the autism spectrum disorder ranges from people who are highly functional to people who need institutional care. So there's a wide range here. Mostly the data that I'm going to be talking about today are data collected from people that are high functioning individuals. What you find with individuals with ASD is they typically have at least normal, sometimes above normal intelligence. So it's not a cognitive problem in that sense. They don't typically have memory issues. They do have some face recognition issues, which I'll talk about later. But in general, their memory, short-term memory, long-term memory seems to be intact. Their visual perception also seems to be very acute. They're very, very good at visual detail and in visual search tasks, things like where's Waldo kind of task. They're extremely good at it. So it's not a general cognitive problem that they're having. It seems to be specifically related to things having to do with social interaction. And one of those kinds of processes that underlie social interaction is face processing. And in face processing, there's no one given model of how faces are processed, and there's not even any given set of processes that compose face processing. In this particular example, what we've got is we have a perceptual input, which is the face, and part of the face processing is being able to distinguish features in the face, things like eyes, nose, mouth, and so on. And that's a very kind of perceptual part of face processing. But then there's also the recognition. So I met you yesterday, I see you today, do I recognize you or not? And that's another part of face processing. And associating names to that face is another part of face processing. And then you get into the emotional kinds of aspects, like interpreting expression. If I'm smiling, am I happy? If I'm frowning, am I sad? What are my facial expressions actually expressing? And these are the kinds of things that individuals with ASD have problems with. Another aspect of social cognition is theory of mind. And in the most broad terms, theory of mind represents your ability to understand, simulate the other person, understand what the other person's thinking, what the other person's intentions are. And it's sometimes called mind reading in the literature. And again, theory of mind involves a whole bunch of stuff. It involves your ability to interpret what the person is that you're talking to or interacting with is thinking. I see you start to tap your feet and roll your eyes, and I immediately have a sense that you must be feeling bored or annoyed with me or something like that. Classic kind of theory of mind task is what's called joint attention. If you're sitting there and you look to the left, so if I'm talking to you in a party or something, and I start looking over here, you'll eventually follow my eye gaze. And the reason you do that is you think, well, if she's looking at something, it must be important. I wonder what it is, and I'm going to look. And this actually turns out to be a very early indicator for ASD. That is, kids at as early as 2 will follow eye gaze. Kids with ASD do not follow eye gaze. And so it has a lot of things to do with intention and, what is this person thinking? What do they want to do? And that, of course, has an effect on how you respond to them and how you anticipate their next actions and behaviors. And the third component of this is action understanding. The idea here being that a lot of the kinds of social behaviors that we engage in are based on imitation. That is, I see you do something, you shake hands with somebody, and so next time I'm in that situation, I might shake hands with somebody. I see you smiling, I will smile in response, maybe not even consciously, but I am just responding to your behavior. And it also involves things like having to perceive the important activities that are going on at the time, to understand what the relation of those activities are to the potential goals of the activities, and to understand how these things may be interrelated so that I could imitate them if I need to imitate them. And this is my way of learning how to be socially responsive. So these different components are all what we call part of the social brain. And the social brain essentially involves things like the mentalizing, which is primarily represented by the theory of mind, biological form, which is things like the face recognition, biological motion, which would be the action understanding, and affect, which seems to kind of float around through all of these things. It's involved in face recognition and face processing because we need to know what the expressions mean. It's involved in biological motion in the sense that we need to know whether or not someone's frowning and someone's raising their fist is an indicator of angry or happy or whatever. And so these areas have been put together and thought of as the social brain, and certain particular regions of interest, or ROIs, not ROIs, ROIs, are associated with these various components. So you think things like FFA, which is the fusiform face area, which is in temporal fusiform, is an area that has been found to be activated when people are shown faces. So someone asked something about how you make the relation between the activation of the brain and what's going on. And that's how we do it. We actually show people, let's say, a number of images of faces, and we look and we see what area of the brain is lighting up. Well, that area is then associated with face processing or car processing or whatever, or affect. We have people do tasks where their emotional valence is swayed one way or the other. We see what parts of the brain are active during that, and usually see things like insula, amygdala, caudate, that kind of thing. And motion is associated often with STS, or superior temporal sulcus, and this is an area that's usually active whenever any kind of action, primarily biological action, not necessarily inanimate car or bus or that kind of action, but actual biological motion. So we've associated those things over a number of different tasks. Lots of research has associated these various areas with these components. And over time, people have contributed to the literature, and you find that there are specific areas that seem to be associated with specific kinds of tasks. So again, here's FFA, here's FFA, which is fusiform face area, STS, which is the biological motion area, dorsolateral prefrontal is working memory, and these various areas have been associated with various components of social cognition and behavior. Now, what researchers have been doing primarily is trying to associate a given brain area with a given function. And the idea was that if we can associate, let's say, fusiform area with face processing, we've nailed it, there's the face processing area. If we can nail STS with biological motion, there it is, we've got biological motion nailed. But it turns out that as you look at the data and as you do more studies, you realize that the same ROIs or the same brain areas may respond slightly differently depending on the kind of context or task that they're engaged in. That is, you might find that under one condition, insula might be responding to disgusting kind of stimuli. Under another case, it might be involved in craving during addiction. And so you have this sort of multifunctional use of these regions. I mean, they're basically within the same ballpark. You're not going to have, amygdala start processing visual stimuli directly like LOC does, but they do have some flexibility in their role, and that flexibility is due to the context that they're in. So this kind of path has led people to start being more interested in the interaction of the areas rather than trying to identify specific regions of the brain that have specific functions. why not look at how, in a given context, a network of these regions interact and how that region will change as a function of the network that it's in. So what people have been starting looking at is actually looking at these individual cognitive processes and looking at the sort of networks, brain networks that might support those processes. So we have a face processing network, theory of mind network, action understanding network. And those things, it doesn't really matter. These are actually larger areas than they should be, but I just pulled it into an atlas and quickly did it. But what we found is that, and again, this varies from person to person, you have a fair core area that's associated with face processing, a fair set of core areas that are associated with theory of mind, and another fair set that are associated with action understanding. But different researchers will add or subtract various areas. So you can't take it too seriously, but some of the areas that seem to be consistent are fusiform gyrus, which is that FFA area that I was talking about, amygdala, which is associated with emotion, superior temporal sulcus, and superior temporal gyrus, which is that biological motion area, and these things seem to be associated with face processing. You'll notice that in theory of mind networks, you also have some overlap. You also have STS appearing, and you also have amygdala appearing, but you have other areas appearing like TPJ. TPJ is in the temporal parietal junction, and it's often associated with angular gyrus. And this area seems to be identified with what people are thinking about themselves and thinking about other people's intentions. It's the theory of mind area. But as you can see, there's a number of other areas that may or may not be involved in theory of mind processing. And then action understanding, same thing. The core aspect of the action understanding network was based on some monkey work that was done by Rizzotti, who found two areas that he called mirror neurons, because whenever the monkey was looking at something, somebody performing an activity, and the electrodes were in the brain, those particular areas would light up and seem to be responding in imitation of the thing that they were watching. And then a human analog was found in the inferior frontal gyrus and the inferior parietal lobe, but there are other areas that are sometimes involved in action understanding. Okay, so let me tell you about the experiment that I'm going to talk about today. This set of stimuli were designed to tap into the different kinds of networks that I've just been talking about, action, face, and theory of mind. And as with areas, tasks can often involve multiple kinds of networks. So for example, the one that is least likely to involve other kinds of network is the arrow object task. In this task, the idea was to see whether or not the action involved in directionality would be activating certain areas of the action network, and whether that was going to be similar in ASD subjects and in neurotypical subjects. And that only, the arrow object task has a pretty much fixed role for tapping into the action network. However, you get down to, where's my cursor? You get down to the eyes left right. Turns out that this eyes left right task, which is they're asked to determine whether the eyes are facing left or the eyes are facing right. This task is the hardest task for individuals with autism to do. They do very poorly on this task. It also is a task that taps into all three networks. So it's used for action understanding, it's used in a theory of mind, because this is like the joint attention task. I'm gazing somewhere else, what am I doing? And it's also involved in face processing, because it's a face that I'm looking at. What they did is, this is a sample trial. What they had is, they have a fixation. They were told ahead of time before a block what their target, what their task was going to be. So in this case, the task was, you're going to see an object, and what I want you to do is, when you see the next slide, tell me if the arrow or the person is looking at that object. So in this case, the answer, the target was clock. It's pointing at the clock, so the answer would be yes, and they were given a reminder of which finger to use for yes and no. And all the tasks were answered yes and no, although they might have been stated slightly differently. The data was collected in a Siemens scanner, and in this case, what you see on the left is the person who's lying prone. They lie prone, this area right here, if you Can you see that? That's a mirror that's attached to the head coil. So as the person's laying in the scanner, there's actually a back projected screen behind them. So they're laying there, they're looking up at this mirror, the mirror is reflecting the screen behind them. And then they're given a response box where they can respond yes or no. And it's a very simple, easy task for subjects. So if you collect the data, I've broken this up into the, this is GLM activation based on FSL. And I've broken it up into the face, TOM, and action networks, and for various contrasts across tasks. The blue is the individuals with ASD, the yellow is the individuals that are neurotypical, and as you can see, for example, what you're getting is activation of fusiform here for face in the neurotypicals, not so much for the autistics. Now, the thing for the purpose for us is this graph analysis, and I used IMaGES, and this is the interface for IMaGES. It's a very nice, Joe, where are you? It's a very nice interface. I love using this tool. It's very intuitive, easy to use, and produces very robust findings very quickly. So there's your plug. And the way this works is that we do extract the time series, and somebody had asked a question about that earlier. Each area that we're interested in looking at a connectivity graph for, we pull out the time series associated with that ROI or that area, and we feed it into IMaGES. Now, there is some variance on how you pull out the time series. You can either pull out the mean time series for the area. So if you have an ROI that has, let's say, 100 voxels in it, you might just get the mean time series for that area. You could choose the most active voxel, that in the belief that the most active voxel is the one that's responding most to that stimulus, or you can just let's see. I guess those are the two main ways of doing it. What I did in this one, I took a mean time series from each of these regions that we were interested in. The regions were selected as a function of the theoretical basis for each of these networks. So like I showed you in earlier slides, there were areas associated with face processing, areas associated with TOM, areas associated with action. So those became my regions of interest, and for each of those networks and for the given task, I pulled out the time series and then fed that into IMaGES. And what you do is you have a number of different subjects. So as Joe said, this is a multi-sample or multi-subject approach, which allows you to actually aggregate over all of your subjects. And there seems to be very little limit on how many subjects that you can aggregate over. We've done it over up to, I think, 20 or 30 and never had a problem. It was still pretty fast and pretty nice. And when you take all these subjects and you aggregate them, you end up magically getting this connectivity graph at the end. So let me tell you what we found using this approach. Here, this is for the face network. So all of these tasks were considered to be targeting or eliciting activation from the face network, the ELR, which is the eyes left right, the EOC, which is eyes open closed. Subject's task was to decide whether or not the picture of the person had their eyes closed or had eyes open, and mouth open and closed for the same kind of thing. Was the mouth open? Was the mouth closed? Yes, no. All of these were yes, no. So these were all things tapping into face. And as you can see, very little difference between the autistic subjects and the NT subjects. Now you have to remember that each of these networks has their proponents of what's the cause of problems in autism syndrome. And everybody says, no, it's got to be face processing. No, it's got to be action understanding. No, it's got to be theory of mind. And most of these proponents are very particular in the network they're using. So what this task was aimed at doing, the study was aimed at doing, is actually taking those different networks and putting them head to head and seeing, are they all affected, or some of them more affected than others? How, what exactly is happening? Well, looking at this, we see the face network, at least for these kinds of tasks, was not affected. The individuals with ASD did not differ significantly from those with that were neurotypicals. However, you look at the TOM network, this is the theory of mind network, you say very, very different the edges are missing, the edges are in different orientations. It's completely different. The other thing to note about this is that whereas the neurotypicals were very stable in their pattern of connectivity across the two tasks, EO and ELR, that were tapping into the TOM network, the autistic subjects were very different across those two tasks, as well as being different from NT. So these guys are responding very differently, not only in general from neurotypicals, but even across the same tasks that are meant to tap into that network. Similar kinds of things in the action network. What you see is, what's interesting here, in the AO condition, which is an arrow, has nothing to do with faces, there's no social cognition involved in here, it's purely an action directional kind of thing. The individual with autism perform just as well as the people with the neurotypicals. And in fact, I have behavioral data for this too, so I have RT and I have accuracy data and all this stuff. And this is consistent with that. They were also, that was the best condition that they performed in. They were the most like neurotypicals in this particular condition. And the connectivity patterns are similar. Now, if you get into other kinds of tasks, these other tasks like EO, you get an extra edge here for the autistics, you don't for the neurotypicals, and then you're missing an edge here for ELR. And ELR was one of the hardest tasks for subjects to do. They performed the slowest and the least accurate on this task, at least the individuals with autism. So what I want to do is kind of emphasize these differences that I'm talking about. If you look at the face network, I've picked one task, that task that was tapping into all three networks. And if you look at the face network, you see that there's really no difference between the individuals with autism and the individuals that are neurotypical. For the TOM, you get a big difference, and for the action, you get a difference. So even though this is the same task, it's eyes left, right, are the eyes facing left, are the eyes facing right, depending on the context or the task that they're doing, or depending on the network that they're tapping into, will determine whether or not that network's functioning. So for that particular task, it's functioning perfectly fine for face processing, which indicates there's really not a deep problem with face processing, at least for these tasks, but there is deep problems with theory of mind and with action understanding. So basically, this is more of a high-level summary. You find that the face processing is not very different between individuals with autism and individuals that are neurotypical. However, both theory of mind and action understanding seem to be very different, respond very differently as brain networks than it does in neurotypical subjects. And the effect of connectivity, or the network connectivity for the individuals with autism, tends to be less stable across tasks that are tapping into that same network than it does for neurotypicals. Neurotypicals tend to be very stable. You have three tasks tapping into a network, all three graphs look the same. With the individuals with autism, those three tasks may produce very different network responses and network connectivity. So in addition to doing the work with the autism, our labs has also been involved in other kinds of research using IMaGES to understand the brain networks underlying various kinds of cognitive situations. Most of these are not clinical. There's one that's clinical, I think. So one of our graduate students right now is doing preference decisions. And in the preference decisions, she's asking subjects questions like, would you rather go skiing or would you rather go bowling? Okay, and she has these lists of preferences pre-test of what subjects like to do. They're bowlers or they're hikers or whatever they are. And she has them, and she presents these in two different contexts. And the one context is called positive framing, and what she does is she said, which would you rather do skiing or bowling. And the negative context is which one would you rather not do? Okay, so it's a very simple kind of manipulation, but it turns out that that manipulation has a big change in the connectivity of the brain areas. So in the positive framing, caudate is not involved. In the negative framing, caudate is involved, and caudate is associated with things like negativity and unpleasant kinds of emotions, which makes sense. Okay, another thing is language processing. We had a graduate student who was studying bilingualism, and she was looking at Spanish speakers and English speakers of various levels of ability, and when they had started learning the two languages, did they learn it English later on, did they learn English simultaneously, et cetera, et cetera, and a lot of different variables associated with that. The key thing here is that what she's discovered, which is really cool, is that she replicates what was a theoretical model, Coulthard model, indicating a dual root for bilingualism. And that had never been shown before, and it was just theoretical, and it was just based on functional kinds of stuff. And she actually got it back out from IMaGES, from using it. And we looked at it and we went, whoa, this is bizarre. So, you know, another reason that we believe that IMaGES is actually doing something valid and useful for us. Cocaine drug usage, this is something I'm collaborating on right now with someone. And basically, these are people who are cocaine addicts and people that are not addicts, and they're presented with stimuli that are either drug-related, so they see pictures of cocaine or, I don't know, cocaine paraphernalia or whatever, and they see other pictures of just nature scenes or pretty objects and things like that. Those are the neutral things. And what you find is that you get a very different, I don't know, you probably can't see it because it's too small, but there's the theoretical addictive network, which goes through the memory and through amygdala and back into the frontal areas. That is captured here for the cocaine users, and particularly for the cocaine users looking at cocaine stimuli, than it is for the non-users. And finally, another project which I don't know a lot about, but I was a consultant on, again, just for the data analysis, and that has to do with male and female orgasm or sexual response. And this was part of a poster that they had presented at SFN, Society for Neuroscience. Apparently got a good rave. I think people like to see studies about sex. But essentially, what they found is that the areas are differentially, of course, activated in both males and females, and that in particular, the female sexual response tends to be much more complicated than the male sexual response. Big surprise, right? Big surprise. But it's science. I'm sorry?
Did you use the Venus and Mars algorithm?
No. Okay, so this isn't a problem list, but it is a wish list, and it's things having used this in various kinds of contexts. And I am, again, an experimental psychologist. I am not a statistician, so, you know, I don't know how well this translates or whatever. But one of the things for psychologists is determining how to make comparisons across various graphs. So I showed you comparisons between autistic individuals and neurotypicals, or between users and non-users and so on. And I told you, you know, there's a missing edge here, or there's an orientation change there. But I have no way of quantifying those differences. I can't tell you, is this 50% different from the other one? Is it 30% different? How different are these two graphs? And if I were to do it in another kinds of context, using another kind of graph application, would I be able to compare the results I got from that with the results I got from IMaGES? So this comparison thing is kind of an issue that I would like to see resolved. Means of assessing the model validity. This has to do with kind of an anal thing on psychologists' part of statistical significance. So most of the things we do, you know, it's significant at the 0.05 level or 0.01 level or whatever, and we have this, you know, inferential statistic that we can use to put the rubber stamp of approval on the data that we have. And there's nothing quite the same here. You have a BIC, or you have a goodness of fit measurement for that graph, but it doesn't really tell you whether or not the graphs are accounting for 90% of the variance in the time series, 80%, 20%. How good, we know it fits it well, but how good in terms of an absolute sense of understanding the data? And again, you know, can you, often when we come up with these graph solutions, we get this family of the MEC, right? Family of equivalent kinds of graphs. And the BIC scores don't differ a lot. The goodness of fit scores don't differ a lot. So how do I choose this one versus that one? I mean, is a BIC score that's, you know, 10 points different from the other one, is that a significant difference? Is that something that should push me into choosing that one, or does it not really matter? So it's mostly those kinds of issues that concern people like me who are trying to use this in this kind of setting. So I'm going to end, and I'm going to thank Steve, Hanson, Joe, and Clark, who were our co-authors on this paper that just came out in Brain Connectivity. And I'll let you ask me questions. Yes?
For your problem with the graph comparisons, have you considered doing a kind of permutation test?
No, again, I'm a really low-level cognitive psychologist. I mean, this is, you know,
The easy thing would be to shuffle the groups, right? You have autism people and non-autism people. You just shuffle them randomly and estimate the graphs and see how much they differ. And you do that many, many times.
But again, it's the how much they differ part. Like, how do I quantify that difference?
You just count how many edges or whatever you want, and then you get a null distribution of what you would expect if there's no real difference between the groups.
Right.
And then you compare what you saw on the real groups to this distribution, and you get your p-value also.
That's actually a good idea. I'll try that. I still have.
Isn't there a problem with the number of subjects? I mean, so if we only have something like 10 subjects. Let's say we have 5 autistics.
No. There were 9 and 9.
Well, then I'm just giving.
Yeah.
Well, that's maybe.
So then I'm going to do that permutation test 1,000 times or something?
Yes,
is
you're
small
doing that, and
sample?
your power might not be super.
Right. So the
But it
uncertainty's
would
still going to
be
high.
statistically valid.
So there it'll be high. Sure.
No, that's actually a really good suggestion. Thank you. Because that's always a big issue. Is how do you quantify that difference, or how do you quantify how good this thing
It's
is?
the most straightforward way I can think of.
Thank you. Anything else?
As somebody who also studies individual differences with imaging, I realized that in your autism experiment, there are, in terms of background knowledge, there are 2 causal manipulations. There is the experimental manipulation that you do with your input, and there's also the naturalistic experiment of how these people have normally developed because of things. We don't know the causes, but clearly there was a causal factor that permuted their brain.
Whether it was genes or environment or whatever, right?
So you looked at the effect of your experimental manipulation, but I can also imagine that we could take, for example, the same ROI data and get the regression coefficients, for example, for the effect of your experimental manipulation, which would presumably reflect how well the brain processes the information that.
Well, it's not necessarily how well it processes. It's whether it's recruited or not.
Right. To what extent it's recruited by that information. And you could also build a causal model where every subject would be a case. And I'm wondering if one model could be used to constrain the others and to what...
Well, in some sense, I think that's what IMaGES does, right, Joe?
Yeah.
Can you elaborate? Please? Please, sir? It's, okay. The idea is that you have these different subjects, and can you use, like in some kind of cascading model, a way of constraining where the model goes as you go across the subject sets? And I'm saying that's basically what IMaGES does. It's self-constraining, right? It uses the other data from the other subject population, or the
Calculating averages of PIC scores. So in a sense, it's using the data from all the subjects.
But are we using the group membership variable, for example? Could we use it as a potential causal factor?
You could. Why not?
So IMaGES would allow us to do that?
Well, I mean,
I don't know how you would
the question
feed
is
it in.
whether IMaGES currently does that or whether I could add it. Unless you do it wrong.
And what's the answer to the question? The answer is yeah, sure he could.
There you go. Yes.
So if I understood correctly, you used these two groups. You said that the subject group were high-functioning autistics.
Yeah.
So if you go along the spectrum further and further,
You should find even more disruption. Maybe a drop out of edges. I mean, part of what needs to be done now is having seen that you have orientation differences or you have edges dropping out, whether or not as you get into more impairment, does that number of edges drop or does the orientations change dramatically? What exactly is happening? And that's exactly what you'd want to do. And you want to see that across the various networks. So what thing seems to be most impacted by the impairment continuum as you get
and
worse?
when you're talking about the variability, talking some general measure over the entire graph, but presumably some edges are less or more variable than others.
You mean
Some edges
over subjects, or yeah? I mean that's also an issue that I didn't bring up, but we've been talking to Joe offline about, is producing some kind of generalized graph right now. What I do is I feed it in and I try to, all the edges obviously are significant that are appearing in the graph solution, but what we wanted to do is have some generic graph for that group. And right now what IMaGES does is it gives you, it's a generic graph, the edges and the orientations are there, but the regression values and so on are different per subject. And what I want one kind of generic graph that represents the group. Again, because of this psychology thing of, we're doing group stuff. Some people do individual studies, but, we want to know what the average, if I was doing to grab an ASD out of the air, how would this person respond? And you need averages for that kind of stuff. Yes?
I saw that you run a structural equation modeling, or you try to estimate the coefficients in your graphs. Does that have a biological interpretation, those coefficients?
That's still under debate, okay? I mean, this kind of work, this is the first stuff that's ever been done using effective connectivity for any kind of psychological thing. Most people use just functional connectivity. They put a seed into one of the areas and they see what other areas are correlated with it and whatever. So so there's still a lot of questions and issues. Like exactly, what does this regression mean? Does it mean, if you have a negative value or if you have a low value versus a high value, does it really reflect the strength of that edge? I don't know. Yes, Glenn.
What does psychologists mean by functional?
It works.
A serious question. I just don't understand.
This is just a terminology that's used in the literature to differentiate the kinds of graph analysis that's done. And with functional, it means that there's no causal implications. You're not saying that A causes B or A precedes B or whatever. All you're saying is these things are correlated. And it's not my choice of terms, but that's what appeared in the, and then effective connectivity was differentiated by that because effective connectivity tells you something about the precedence of one activated node before another. That's just terminology.
So it's really just not wanting to say up front that it's just correlation?
Of course. When you can use jargon and you can sound important, don't you want to do that? IMaGES, right? Anything else? Yes.
I was wondering, like in the autism study, it seemed like you could have made your point just with adjacencies and not with orientation. So I was wondering if, as a psychologist, you thought the orientations you were getting were plausible relative to...
Yes. Yes, the orientations actually are important. If I was only interested in the adjacencies, then I could use functional connectivity, and all I'd care about is that these things were related to one another. But what I do really care about is whether or not some of these models that I showed you earlier of the way the information is progressing from one ROI to another, can I capture that with the connectivity analysis, and does that graph actually make sense vis-a-vis anatomical data or other kinds of data that's established a certain pathway. So you're not going to have things going I'm not going to say that because you do have some of that, but there are certain paths and path forwards and whatever that you're expecting to see, and some that you would probably not expect to see ever. So there are constraints on it in that way, but since we don't have that much information about this, we're still kind of grasping around in the dark. You make your best guess. And so what I've tried to do is when I've used IMaGES, is try to be consistent with whatever the anatomical data or other kinds of literature seems to suggest the edges should be like in orientation. Anything else? Okay, thanks.